Swayam Prabha. Digital India. Educated India.
Hello everybody, I'm Dr. R. Vijayalakshmi, a molecular oncologist from Cancer Institute, Adyar. We are going to be doing a course on precision oncology, and today's topic is going to be tumor microenvironment. I have given my contact email here, so whoever is having any doubt or any suggestions can write to me, reach out to me through this email ID. So let's start the topic. So this lecture is going to be divided into two parts, part 1 and part 2. In part 1, we will be dealing with what is tumor microenvironment, what are the components of tumor microenvironment, how tumor microenvironment interacts with the tumor, how the tumor progression is dependent upon the tumor microenvironment. At the end of the lecture of today, you will be able to understand these topics, and as a part of part 2, we will be covering how this tumor microenvironment can be therapeutically targeted. So let's get into the topic. Tumor microenvironment is the environment around a tumor. It's as simple as that, which includes surrounding blood vessels, immune cells, fibroblasts, signaling molecules, and extracellular matrix. Now, if you see the cartoon here, I have shown a house, and around the house, what is it that you see? You see a fence, you see a garden, you see some flowers. So, this fence, garden, and flowers is similar to the tumor microenvironment, where the house itself is like a tumor. Cancers have very complex ecosystem which comprise of different cell types and non-cellular factors. So this tumor microenvironment is otherwise called as the tumor stroma. This tumor stroma is a critical component of tumor microenvironment, and it has got very crucial roles to play in tumor initiation, progression, and metastasis. Now, take a look at the tumor microenvironment shown here. So, you have the orange cells here are the malignant cells. The gray ones here that I am showing you is the necrotic and the hypoxic cell. And then you have the tumor associated neutrophils, the dendritic cells, the tumor associated macrophages, the myeloid derived stromal cells. So, these three, the dendritic cell, the tumor associated macrophages, and the myeloid derived stromal cells come under the category of myeloid cells. And then you also have lymphoid cells, the lymphocytes, that is the T lymphocyte, the B lymphocyte, the NK cells, they all come under the category of lymphocytes. And then you have, in addition to these cells, you have the cancer associated fibroblasts, and they are the CAFs, and the mesenchymal stem cells, which can leach into the blood circulation. So if you see, a tumor microenvironment is comprising of all these cells, and then there is a lot of interaction which is happening between the tumor cells and the other cells, other non-malignant cells which are present in this microenvironment. And that is what we are going to be understanding in today's lecture. This tumor microenvironment is very important for the cancer progression, and they help in immune evasion, tumor escape, hypoxia and acidosis. This we will be seeing in today's lecture, and the metabolic exchange, invasion and metastasis, which is kind of paved way by the tumor microenvironment, the concept of angiogenesis and tumor growth. How these things are happening in tumor microenvironment is what we will be seeing in today's class. So as we saw in the previous slide, when the cancer has to dislocate from its primary location and travel to a different site, and this aspect of cancer is called as the metastasis, and that is what makes this disease very lethal. So when the cancer cells get into the circulation And when they travel to a distant site, they require what is called as the microvasculature. That is, they need new blood vessels through which these cells can commute to a different site in the body. So the tumor microenvironment is basically the site of this microvasculature. There are several cells present here. You have the endothelial cells, the pericytes, the angioblasts, the endothelial progenitor cells, fibroblasts, mesenchymal stem cells or stromal cells, and other stromal elements. All these cells form the tumor microenvironment. And then these cells will be secreting certain growth factors and certain chemicals which are called as the pro-inflammatory cytokines. So the pro-inflammatory leukocytes, which are the immune cells, the lymphocytes, the tumor-associated macrophages, they are all kinds of cells, kinds of leukocytes, kinds of WBCs which release a certain pro-inflammatory cytokines. That is, they will cause inflammation. So all this surrounding the extracellular matrix form the mechanical scaffold. So the whole thing is like a mechanical scaffold, and that will define the tumor elasticity. So the cancer progression involves the tissue elasticity, and this is very dynamic and it keeps changing through time. The tumor cells metastasize through the leaky tumor vasculature and they migrate through the blood vessels to the other organs. And this is what we call as the metastasis. Disruption therapy may break down the physical barrier, that is, the extracellular matrix mechanical scaffold, leading to imbalance of the cancer ecosystem. So, whenever we treat a cancer cell through any kind of a modulation, any kind of either chemotherapy or radiation, what exactly happens is this is going to cause an imbalance in the cancer ecosystem. So this is Stephen Paget in the year 1889 came up with the hypothesis which is called as the seed and soil hypothesis. That is, tumorigenesis, cancer progression and metastasis. They are all strongly dictated by cell autonomous genetic and epigenetic changes. That is, the tumor cells as such will be having a lot of mutations which is going to make them autonomous. They will stop responding to the growth factors and growth factor signals basically. They will stop responding to that and they function in an autonomous way. But the tumor stroma interactions are the ones which is very important for a sustained progression of cancer. The cell may have its own mutations, but then for a particular cancer to progress in a sustainable way, there has to be very successful tumor and stromal interactions happening. This is what is called as the seed and soil hypothesis. So, Stephen Paget suggested that when a plant goes to seed, its seeds are carried in all directions, but they can only live and grow if they fall on a congenial soil. The soil has to be in a way that it is going to help in the growth of the seeds. So, while many researchers have been studying the seeds, meaning the tumor cells, the properties of the soil, that is what he means is the tumor microenvironment, may reveal valuable insights into the metastatic peculiarities of cancer cases. Paget's hypothesis is staying true to this day. Paget's description of the ability of cancer cells, so they are your seeds, to grow only in certain secondary sites depending upon their microenvironment or the soil still remains accurate. That is, when the cancer dislocates from a primary location and gets metastasized and moves to a different site, that site, the decision of moving to a particular site is dependent upon a very congenial tumor microenvironment in that site, which can support the growth of this migrated cancer cells. Now, this is a non-malignant tissue that you see here. So these are the epithelial cells which are very nicely orderly arranged, and you have the cell adhesion junction which are intact, and this is your basement membrane, and all this component below the basement membrane is the stroma or the tumor microenvironment. And as I mentioned, there are several kinds of cells shown here. So the non-malignant epithelial tissue is supported by a stroma. So what you see here is a stroma, and it is composed of the extracellular matrix. So what you see, this pink one here, the background is basically your extracellular matrix. And then you have the fibroblasts here, and then you have the mesenchymal stromal cells. And if it is going to be a bone tissue, then you will have osteoblasts. And if it is going to be a cartilage, then it will be chondrocytes. So the tumor microenvironment's composition also will change depending upon the organs. So the cells in the non-malignant stroma will be normally in a quiescent state. That is, they are perfectly balanced and they have a kind of homeostasis. In the ECM, that is in the extracellular matrix and the epithelial compartment, if you see, everything is very, very orderly, and the extra proliferation or extra motility or invasion is negatively regulated. So therefore, the whole thing is under balance and control. Whereas this is what happens in a tumor situation. Now, if you, in case of a tumor, you see that the basement membrane is breached and the tumor cells are getting into the stromal component. They are invading deeper, and you can see some stromal cells coming, some tumor cells coming into the stromal region here. So when cancer develops, the stroma undergoes vast changes to become fibrotic and activated. So the extracellular matrix becomes denser and more rigid. There is a lot of change which happens in the tumor microenvironment when the cancer is starting. So the extracellular matrix becomes denser and more rigid, and it is composed of alternative forms of connective tissues like tensin and fibronectin. So these are some of the connective tissue proteins that will be coming up, and this will help in the cancer cells to invade through the stroma. So, and the fibroblasts and the mesenchymal stromal cells will change their shape, and they will also change their expression profiles in a way that they become more proliferative, and they secrete higher levels of growth factors like cytokines and chemokines. So you see that the fibroblasts and the mesenchymal stromal cells are undergoing a lot of changes in a way to support malignancy when the malignancy actually starts. So the stromal fibroblasts in the tumor microenvironment are referred to as the cancer associated fibroblasts. That is what we call as CAFs or the myofibroblasts. So once the stromal fibroblast is getting converted to cancer associated fibroblasts, then the kind of cytokines and the kind of chemokines that will be secreted by them will promote tumor formation. The tumor trauma promotes cancer progression and metastasis and leads to resistance to therapy and disease recurrence. That is why the tumor stroma is very, very important in cancer therapy. Now, the tumor stage basically depends upon the stromal activation. That is what we are seeing. Now, as I mentioned, this is a normal epithelium and this is a pre-malignant dysplasia, and this is a scenario of carcinoma. That is when the carcinoma is starting, So what actually happens is the stroma becomes reactive in nature. Now we will look at this little more closely. Now what you see here, these are your keratinocytes and these are your basement membrane and this is your stroma. So you have the collagen bundles, you have limited number of fibroblasts, and then you have the capillaries, that is your through which the blood supply is there. You have the granulocytes and the monocytes and macrophages. So they are all limited in number and they are in the state of equilibrium. But then in the pre-malignant stage, when the epithelial cells start proliferating, this is what we call as the hypoplastic epithelium. So the stroma, when the epithelial cells are growing in numbers, this happens because the differentiation program in the epithelial cells get altered, and because of this, there is a lot of proliferation that we see in the epithelium. This rapid proliferation in the epithelium leads to activation of stroma, and this leads to activated fibroblasts. So the fibroblasts which were in the quiescent state now get activated. These activated fibroblasts are very important to promote tumor. So what happens is, when these activated fibroblasts are going to be secreting pro-inflammatory cytokines, and there is a lot more changes which is happening, which we will be discussing subsequently, the stroma kind of becomes reactive in nature. When the stroma is reactive, you can see that there is a rapid proliferation of lot of cells. You are having a lot of new angiogenesis happening, that is, new blood capillaries are formed here, and then you have lot of the immune cells which are dividing, and then there's a lot of pro-inflammatory cytokines being released. There is a breach in the basement membrane, and then you are having the cells kind of invading into the stroma. So therefore, the stage of the tumor basically depends upon how much the stroma has got activated. So let's go into components of the tumor microenvironment and their functions in little more in detail. We will be looking at the individual components. So first thing, that is endothelial cells. Now what these endothelial cells do? The endothelial cells provide nutrients for tumor growth, and they constitute roots for metastatic dissemination through angiogenesis, and they contribute to resistance to chemotherapies and radiation. So these endothelial cells are important for blood vessel formation, through which these tumor cells can travel through those new blood vessels to a distant site. So therefore, they are going to be supporting metastasis, and they also contribute to resistance through chemotherapy and radiation. When the treatment is given, there is a concept of radiation and chemoresistance which takes place. So endothelial cells are important. Pericytes also contribute to angiogenesis, and they confer resistance to anti-angiogenic therapy. So the next type of cells that you see in the tumor environment is adipocytes. The adipocytes support cancer mainly through secretion of growth factors and cytokines, and they have also been shown to have role in resistance to chemotherapy, radiotherapy, hormone therapy, and other targeted therapies. Immune cells influence pro-tumorigenic phenotypes. That is, there is something called as the epithelial to mesenchymal transition. We call this EMT. So we will be seeing that shortly. So this is a kind of conversion into a pro-tumorigenic phenotype. So it gets into an EMT. It kind of, from epithelial type of a cell, it gets converted into a mesenchymal type of a cell. That's what is EMT. And then angiogenesis. Angiogenesis means forming new blood vessels and therapy resistance, as well as anti-tumor phenotypes for immune surveillance through diverse and complex mechanisms. So the immune cells can be pro-tumorigenic or anti-tumorigenic, and both kind of phenotypes will be seen, and there has to be a balance between the two. So if you are having an excess of pro-tumorigenic phenotype, then it is more towards cancer promotion. And then the extracellular matrix, which provides structure and support for the cellular components in the extracellular space of tissues and organs, and they contribute to paracrine cellular signaling. Now if you see here, this is an example of the extracellular matrix. So the extracellular matrix basically comprises of interactions of fibrous molecules. Then you have the proteoglycans, and then glycoproteins, glycosaminoglycans, and other macromolecules. So if you see here, you have the fibronectin here, and then in this fibronectin, you are having the proteoglycan complex. So this proteoglycan complex consists of, this is a polysaccharide backbone. Along with it, you are having proteins and carbohydrates put together, and this is your collagen fibers. And then you also have, this is the plasma membrane, and you have the integrins for interactions, and there are also microfilaments of the cytoskeleton. So therefore, the extracellular matrix basically has two main compartments, the basement membrane. So this is your basement membrane and the interstitial ECM. So the ECM is slowly and constantly degraded by enzymes such as collagenases and matrix metalloproteinases. So when the ECM gets degraded, the cells can start moving from their location and they can enter into the circulation. So the cells have to basically breach into the stroma and they have to travel through the stroma and reach the blood vessels, and then they have to get into the blood vessels and start moving. So therefore, for this particular process, you need certain enzymes which can degrade your extracellular matrix, and so matrix metalloproteinases are chiefly involved in degradation of the extracellular matrix. And collagen is the most abundant protein in the interstitial extracellular matrix, and apart from collagens you have elastin, fibronectin, tenascin and laminin. All these proteins have critical roles within this compartment of ECM. So we will see one after the other. So one of the most important player is fibroblast. Now in this particular section here, you see something which is very spindle shaped in a fusiform kind of a structure. So these are your fibroblasts. So fibroblasts are very important, and what you see here, these are your keratinocytes, and this border like thing that you see is your basement membrane. So this is just a histopathological section to show you the connective tissue part. So you have the collagen fibers, eosinophils and fibroblasts seen here. The fibroblasts build the extracellular matrix by secreting collagens and other fibrous macromolecules, which also degrade this network by releasing these proteolytic enzymes which I mentioned. That is the matrix metalloproteinases, which enables increased cell mobility through the extracellular matrix. The interaction between the fibroblasts and the extracellular matrix or the surrounding cells is mediated by the integrin signaling. So you have the integrin receptors through which these interactions happen. The tissue wounds or certain pathological conditions can activate fibroblasts. So we saw in the previous slide that how a quiescent fibroblast gets activated during a process of transformation or even in the process of wound healing. So anything which is disturbing the equilibrium, first thing will be activation of fibroblasts. So fibroblasts gets activated in such a way that they proliferate and secrete higher levels of protein in an aberrant way. Fibroblasts also have an important role in tissue maintenance and homeostasis by expressing enzymes from the cytochrome P450 family, which degrades the foreign and the potentially toxic molecules. So fibroblasts can recruit and regulate the leukocyte infiltration and inflammation via secretion of cytokines, chemokines and growth factors. So fibroblasts can secrete a whole lot of these chemicals which can be pro-tumorigenic. Now this is how a fibroblast looks. This is a normal fibroblast, and what you see here is the activated fibroblast. So you see that there is a kind of a change in the shape here. Now as such, if you see, this is a fibroblast, you have a nucleus here, and then this is the integrin receptor here through which the interaction happens with the ECM basically, and then you have within the fibroblasts, you have the fibronectin, the fibrillar, ECM, the collagens are present here, and this is you have the actin fibers and you have the vimentin here inside. So the normal fibroblasts are embedded within the fibrillar extracellular matrix of connective tissue, which is largely consisting of type 1 collagen and fibronectin. So basically, a normal fibroblast will have type 1 collagen and fibronectin. Now what happens if it gets activated? When it gets activated, that is when it assumes or acquires an activated phenotype, then it is associated with an increased proliferative capacity. So once it gets activated, it can undergo a lot of proliferation, and it also has an enhanced secretion of the ECM proteins such as the type 1 collagen, tenascin C and fibronectin. As I mentioned, when you have an excess of this tenascency and fibronectin in the ECM, then it actually helps the cells to invade very easily. So therefore, the fibroblasts which are activated will start proliferating and will start increasing the secretion of these ECM proteins such as the type 1 collagen, the tenascency and the fibronectin, and that contains an extra domain which is called as the EDA fibronectin, and also a protein called as SPARC, which is secreted protein, acidic and rich in cysteine. So these are some of the proteins secreted by fibroblasts which enhances the migration of these tumor cells into the stromal area. Apart from the fibroblasts, you have the mesenchymal stromal cells. So they are basically multipotent stromal cells which are commonly referred as mesenchymal stem cells. So they can very easily enter into the circulation. Now, the mesenchymal stem cells are cells that are able to adhere to plastic surfaces, and they have the capacity to differentiate into osteoblasts, chondrocytes or adipocytes in culture. Because they are stem cells, they can get differentiated into any kind of a cell based on the tissue of origin. If it is bone, it will be osteoblasts. If it's cartilage, it will be chondrocytes, and they can express the cell surface marker CD73, CD90 and CD105, but not any of the leukocyte markers. So these CD73, CD90 and CD105 expressing cells are your mesenchymal stem cells, and they can be identified by flow cytometry if you kind of use these kind of antibodies for these markers. So then the next type of cells is osteoblasts. Osteoblasts are unique stromal cells which are responsible for building the bone, and it is a very highly specialized extracellular matrix consisting of a mixture of collagen and non-collagenous proteins that is subsequently calcified with hydroxyapatite. Because in case of bone, there is it is a completely a strong organ, isn't it? So therefore, there is a lot of calcification which happens with hydroxyapatite, and you have a combination of collagen and non-collagenous proteins. In case of chondrocytes, chondrocytes is also differentiated from the mesenchymal stem cells, and they are a major stromal component, and chondrocytes produce cartilage, the specialized ECM which is present in the joints and other cartilaginous tissues. So this is how the ECM composition changes based on the organ. So this is a normal connective tissue. So here we see the different types of ECM based upon the different types of organs. So this is a connective tissue. So you see that this connective tissue, the main function is connection, support and nourishment. So the ECM favors these activities. And then this is cartilage. In case of cartilage, it is more for mechanical support, for viscoelasticity and lubrication. So therefore, there is more number of aggregants, there is more number of hyaline, and then you have elastins which will give you the elasticity of a cartilage. So the point is the ECM composition will vary as per the organ. So this is in bone. Now if you see in bone, you will have more of hydroxyapatite, then you have osteocalcin. So these are more, they give the tissue more durability, strength, a stiff structure and low elasticity in case of bone. And in case of cornea, it has to be more transparent, more translucent. So you see that the corneal ECM is kind of composing of the corneal stroma, and then you have the collagens, lesser number of collagens, then you have certain laminins, integrins. So the constitution is kind of in a way which favors the function of the organ. Now, this is where I'm going to be showing the similarity between wound healing and transformation and malignancy. So, in case of a wound, now what you see here is whenever there is a wound, in the first phase of wound healing, you see that you have the activated fibroblasts, and a lot of immune cells are also getting activated. So all these activated cells are traveling to the site of the wound. Now, in the second phase, when there is proliferation taking place, you see that the new blood vessels are being formed, and these new blood vessels will increase the blood supply or more oxygenation into the area to facilitate healing. And then when this happens, then there is a lot of remodeling of the wound healing happens, where you have the more number of blood vessels being formed, and then there is a lot of fibrosis which happens here, more number of fibroblasts, and then you have more number of collagen deposition in this area, which tends to lead to remodeling of this wound. Now, in case of cancer, something similar only happens. Now, in case of the first step, whenever there is an injury or whenever there is a breach into the area of basement membrane is what is considered as injury, then you have the recruitment of the fibroblast to the tumor site. Then after that, the next stage is the neovascularization. Neo means new, vascularization means formation of blood vessels. So you have the neovascularization where the new cells are getting into the circulation. You will have the blood supply increasing to the area of injury or area of transformation, which will facilitate the it actually is getting converted into a pro-tumorigenic environment. And then when that is completely pro-tumorigenic, and after the cells are completely breached into the basement membrane, they are breaching into the stroma, eventually the breached tumor cells enter into the circulation, and then through the circulation they reach the lymph node. So therefore this is what is metastasis. So the formation of cancer and the concept of wound healing have a lot of similarities. Now, this is what is seen in case of transformation of a normal breast tissue to an advanced invasive ductal carcinoma. Now, what happens in a normal breast tissue? So, you see that the basement membrane is intact, and then this is the ductal epithelium. So, this ductal epithelium and the underlying myoepithelial cells, they are all separated by the basement membrane and from the surrounding connective tissue. So, there is a proper demarcation between the cells and the tumor microenvironment, and then it is there is a basement membrane surrounding the connective tissue by fibrillar extracellular matrix, the capillaries and the fibroblasts. Now they are all very orderly. Now what happens in case of ductal carcinoma in situ, that is DCIS, where the in situ means within the tissue. It is still not breached into the basement membrane, but there is a cancer formation which has got initiated. So in case of DCIS, the lumen here what you see, the blue cells, they are the carcinoma cells. They have started proliferating, and there is a proliferation of the transformed epithelia. So therefore you see that now once this is happening, then all these fibroblasts have become activated. And so you have an increased number of fibroblasts and then increased number of blood vessels as well, which will start supporting this dysplastic cell to grow further. And then during the invasive ductal carcinoma, you see that there's a breach of the basement membrane, and these cancer cells are entered into the basement membrane. That is, through the basement membrane, they are entering into the stroma. So there is a breach of the epithelium here. And this is what is called as the invasive ductal epithelial cells into the stroma. So again you see that the caps are there, and then the blood vessels are also increased in number. But then in case of an advanced cancer, what happens? Here it is only one cell, but then you see that all the other cells in the vicinity has also got transformed, and then there is a breach of basement membrane. In case of advanced breast carcinoma, you have irregular-ness and cords of cancer cells that invade the dense and the fibrous stroma. So this is how the tumor microenvironment keeps changing as the process of transformation happens from normal breast tissue to reaching a stage of advanced invasive ductal carcinoma. So now let us look at the stroma and the tumor interactions and how the tumor promoting activities of stroma happen. Now, as I mentioned, through the increased deposition of collagens type 1 and 3 and de novo expression of tenascin C, they can induce an altered extracellular matrix microenvironment, the ECM microenvironment, which potentially provides additional oncogenic signals, probably leading to accelerated cancer progression. The fibroblasts here mediate an inflammatory response by secreting chemokines such as monocyte chemotactic protein 1, interleukins such as IL-1. So, this activated cancer fibroblasts can release a lot of growth factors. So, here it is shown releasing TGF beta. This is HGF, hepatocyte growth factor. Then you have the VEGF, which will help a vascular endothelial growth factor, which will help in neoangiogenesis, forming new blood vessels. Then it can produce the matrix metalloproteinases, which can degrade the matrix, and then interleukins and all the other cytokines, which will be pro-inflammatory, that is increasing the inflammation in the area, as well as tenascin C, which can help the cancer cells to migrate or to move, to dislodge from their location and migrate into the stroma. So, all this is done by the cancer fibroblasts. The fibroblasts interact with the microvasculature by secreting the matrix metalloproteinases and the vascular endothelial growth factor. Fibroblasts also provide potentially oncogenic signals such as the TGF beta. TGF beta is a very important pro-tumorigenic growth factor and the hepatocyte growth factor to the resident epithelia here, and which can directly stimulate the cancer cell proliferation and invasion, secreting growth factors such as TGF beta and stromal cell derived factor SDF1. So, if you see here, the tumor promoting activities of stroma are multidimensional. On one aspect, the fibroblast recruitment is what we saw, how it can help in ECM production, it can increase in ECM production, and then on other side, it can cause macrophage polarization. That is, the macrophages are attracted to the site of tumors, and then that can in turn lead to increased angiogenesis and also a lot of ECM remodeling. And then there is also ECM degradation and intravasation through secretion of several proteolytic enzymes or the matrix metalloproteinases, which can help in ECM degradation. On the other side, they also carry out immune suppression. That is, using your myeloid derived suppressor cell, and then you have what is called as the T regulatory cells. So, these are also getting attracted to the site of tumor, and they can in turn inhibit the B and the T cell response, and they are suppressing the immunity in a way that the tumors are able to suppress the immune system against activation to avoid elimination. So, they are able to function in a multidimensional way. On one aspect, they recruit the fibroblasts, and then they recruit the macrophages, and then they suppress the immune system and also produce enzymes for ECM degradation and intravasation. So, the whole system is hijacked by the tumor. The tumor in turn hijacks the entire tumor microenvironment in its favor to facilitate its progress. Similarly, in the earlier stages, if the tumor is in the earlier stage, it is also possible to reverse the tumor phenotype by stromal normalization. So, one has to see that the the stromal homeostasis is not lost. So, if we can normalize that, then a reversion is possible. So, suppose you have a situation of a reactive tumor stroma that, as we saw earlier, so you are having these are all your blood vessels, then you have the VEGF receptor, and these are all the proteases. So, if you can treat with say an anti-angiogenic agent, so that can actually stop the blood supply, and when the blood supply to the area is curtailed, then the stroma can become normalized, or it can come back to its original form, and therefore the stroma becomes normalized, and from your from your carcinoma stage, it is becoming like a pre-malignant dysplasia. So, this is when you are able to target the tumor microenvironment with proper agents, so like your anti-angiogenesis agents, then it is possible to restore the homeostasis in the stroma. Now let us look at what kind of crosstalk happens between the tumor cells and the activated stromal surroundings. So one aspect is, now this is your tumor cell. So it will have autocrine signaling, that is, it will release certain growth factors which can influence the cell itself, which is secreting it, that is called as autocrine. So through autocrine mechanism, there can be secretion of proteases and inhibitors, ECM molecules and growth factors. So they can act on the tumor cell. And then it can also have a paracrine mechanism where this particular tumor cell is influencing the neighboring cell. So here that is paracrine to the tumor. So again you can have secretion of growth factors, you can have proteases, secretion of ECM molecules, activation of peptides, cryptic factors, which can in turn lead to proliferation and invasion. So apart from acting on the neighboring tumor cells, it can also be paracrine to the stroma. That is, the tumor cell can influence the stroma which is just next to it as well. That is also by secretion of growth factors, by secreting proteases and ECM molecules. Now this in turn, when it is going to, the tumor cell is going to influence the stroma, then that will lead to angiogenesis, new blood vessel formation. So this is what is shown here. You have the endothelial cell, which is a blood vessel. You have the pericyte, and then they are producing certain kind of growth factors here, so which is leading to new blood vessel formation. Similarly, it can also lead to recruitment, proliferation and differentiation of inflammatory cells, pro-inflammatory cells, which can increase the inflammation in that area. Plus, the tumor cell can also secrete certain factors which will activate the fibroblasts. And we just now saw what activated fibroblasts can do. So, the fibroblasts, when it gets activated, it will lead to mobilization of growth factors and biologically active ECM fragments by proteases, and this in turn can help the tumor cell again to proliferate. So the whole thing happens in a form of in a cyclic way, and there is a lot of simultaneous crosstalk which happens continuously between the tumor cells and the activated stromal surroundings, which decide on the aggression of a particular cancer. So this particular cartoon shows what is necrotic, what is hypoxic, and what is normoxic region within a tumor. Now, as we saw that when there are new blood vessels formed and the blood supply to that area is going to be high, that means that is an oxygen rich area. So that area is called as the normoxic region within a tumor. And then deeper into the tumor, you will have an area where the oxygen cannot reach. So that is called as the hypoxic region, where the oxygen concentration will be low. And then beyond that, there's again another area which is called as the necrotic area, where the cells are dying. Because of lack of oxygen, the cells are already dying. So within a tumor, you have the normoxic, the hypoxic and the necrotic region, and the hypoxic region here is very, very important for a pro-tumorigenic environment. Now, what happens in case of hypoxic area? When you have a hypoxic region within a tumor, that leads to production of a particular protein which is called as the HIF1 alpha. Hypoxia inducible factor 1 alpha. You have the HIF1 alpha and HIF1 beta. There are two types of proteins. So this HIF1 alpha will get induced, and that can in turn lead to formation of certain cytokines, which in turn can lead to a proliferation of lot of genes. It will lead to transcription of lot of genes which are important for metastasis and invasion. All these are induced by this HIF1 alpha. So this HIF1 alpha plays a very, very important role in metastasis, and this leads to increased tumor vasculature. Again, when kind of treatments like radiation is given to a bulky tumor, that also can create hypoxic regions within tumors, and that in turn can induce the HIF1 alpha. So the HIF1 alpha's expression is generally more favoring tumor vasculature. So therefore, one has to take care of this HIF1 alpha protein, and then the relative amount of stroma and its composition may vary considerably from tumor to tumor, but they do not correlate with the degree of the tumor malignancy. So normally every tumor to tumor there will be a difference in the amount of stromal component present, but the interactive signaling between tumor and stroma is very important, and it contributes to the formation of complex multicellular organ. The cancer cells themselves can alter the adjacent stroma to form a permissive and supportive environment for tumor progression. So that's what the cancer cells do. They kind of alter the adjacent stroma in a way that is permissive and supportive for their own growth. Now, how do the cancer cells do that? The cancer cells usually go about generating a very supportive microenvironment by producing stromal modulating growth factors. There are several stromal modulating growth factors which support cancer growth, and these include the basic fibroblast growth factor, members of the vascular endothelial growth factor, the VEGF, and the platelet derived growth factor, which is PDGF, epidermal growth factor receptor EGFR, ligands, that is EGF, and interleukins, colony stimulating factor CSF, and transforming growth factor beta, and others. So there are several growth factors which are produced by cancer cells, and then each one of them have a mechanism to kind of generate a supportive microenvironment for the growth of cancers. But to make things simple, we will just focus on TGF beta to see how the activity of TGF beta helps in tumor growth. Now, the TGF beta signaling promotes fibrosis and progression by the EMT. EMT is epithelial to mesenchymal transition in different organs. Now, what you see here, this is basically an epithelial cell. So, you can see there is a tight junction there. The cells are placed in a particular apical order. So, you have the basal side and the apical side, and all these cells are adhered to each other with the help of these junctions. Now, what happens when there is an epithelial to mesenchymal transition? You are having a cell which is stationarily placed in one place assumes a motile form. That is, in a way, it is getting transformed into a particular kind of phenotype which will favor its movement from one place to another place. So that is the epithelial to mesenchymal. As a mesenchymal phenotype, the cell can start moving. So it actually assumes a spindle kind of a shape, and so that it can start moving into the circulation. And epithelial to mesenchymal transition is basically not just in cancer. It is a normal process in the body during embryonic development, during wound healing, cancer metastasis and fibrosis. So this is a kind of a phenomenon which normally occurs in the body. But in case of cancer, as I mentioned, the whole process is very similar to that of wound healing. There is EMT which helps the cells to metastasize. So during the EMT, you are having the certain genes like snail, twist, zeb. These are the genes which will promote EMT. That is, they are converting the epithelial phenotype to a mesenchymal phenotype. And as a part of cell signaling, you have several growth factors like TGF beta, FGF, EGF, HGF. All these growth factors help in the process of EMT. Similarly, you can also have mesenchymal to epithelial transition. That is also possible. But when the cancers have to dislodge themselves and get into the circulation and move forward, the EMT has to happen. And this important aspect of EMT is promoted by TGF beta. So, the epithelial cells lose their original properties such as the apical basal polarity, cell-cell adhesion, and they acquire a mesenchymal feature such as the anterior and posterior polarity, and then a cytoskeleton that favors migration and invasion. So, as epithelial cells, they are stationarily put in one particular area, But when they assume a motile form, that is a mesenchymal phenotype, then that is favoring migration and invasion. The expression of epithelial markers like E-cadherin. E-cadherin is a standard epithelial marker and vimentin is a standard mesenchymal marker. So normally during EMT, the E-cadherin gene expression is suppressed and vimentin expression is increased. So you will have E-cadherin gene expression suppressed and expression of mesenchymal markers like N-cadherin, alpha SMA, that is smooth muscle actin, fibronectin, and vimentin, all this is induced. So in any early cancers or in any kind of a dysplasia situation, when you have these mesenchymal markers being highly expressed, then that could be an indication that the cancer is actually formed, that is, it's invading, or it is the process of transformation has begun. So some of these mesenchymal markers can be can be helpful in early detection of cancers. Now this is a kind of shows the role of TGF beta in hepatocellular carcinoma. So what you see here, this is a tumor cell. So this tumor cell is producing TGF beta, and this TGF beta itself will act in an autocrine way. It will stimulate the tumor cell here, and this tumor TGF beta will also lead to proliferation of cells and also leading to formation of new blood vessels here. So TGF beta is helping to increase the angiogenesis, and then so it can increase the proliferation, it can decrease the apoptosis, it can increase the EMT, and it can increase the invasion and metastasis. So all these things are carried out by TGF beta. So at the cellular level, TGF beta is inducing proliferation and survival of the HCC cells, displaying a late TGF beta signature, promoting EMT, invasion, and metastasis. At the microenvironment level, TGF beta is basically a key mediator of angiogenesis in hepatocellular carcinoma, contributing to a high vascularization of these tumors. So TGF beta also generates a favorable immune microenvironment for tumor growth. So this is how an important growth factor of an ECM can promote tumor growth. So this is another example of how TGF beta and HIF1 alpha kind of come together. So I have already mentioned about HIF1 alpha in the previous slide. So what you see here is a now normoxic region, like how we saw earlier. This is a blood vessel. So you have the normoxic region here. That means the oxygen concentration is normal here, and then you have the hypoxic region where the oxygen concentration is low, and then you have the necrotic region where the cells are dying. Now, in this kind of a scenario, in the normoxic region, what happens is there is a particular protein called as the von Hippel-Lindau protein, VHL. This is a tumor suppressor protein, and this is it's a ubiquitin ligase. So when the concentration of oxygen is normal, the VHL is, because it's a ligase, it can degrade, it binds to HIF1 alpha and leads to its proteasomal degradation. And this proteasomal degradation can happen only in the normal concentration of oxygen. Now what happens in the hypoxic situation? Because the oxygen concentration is low, or in a situation when the VHL protein is faulty or it's mutated as a tumor suppressor, then this in the hypoxic situation, this HIF1 alpha gets stabilized. So if you have a situation where the HIF1 alpha is getting stabilized, and that can lead to activation of several targets which will facilitate tumor progression, like the HIF1 alpha and the beta will bind to the HIF1 binding regions within the DNA, and they can lead to transcription of several genes which are involved in angiogenesis, in invasion and metastasis, in EMT for in case of survival and metastasis, and then for genomic instability, for metabolic reprogramming, for dedifferentiation, for altering the immune response. So many things are happening because of the stabilization of HIF1 alpha. So as I said, HIF1 alpha stabilization can happen because of hypoxia, but then it is also possible to have an increased HIF1 alpha because of TGF beta. Now, Here, that is what is shown here. Fibrosis occurs in several organs as an outcome of wound healing following a recurrent tissue injury. Now, it is defined by the dysregulated production and excessive accumulation of collagen-rich ECM as well as replacement of the normal functional tissue with a fibrotic tissue. This is what we see in an oral pre-malignant condition called as oral submucous fibrosis, where we have an excess fibrosis in the oral area. So, whenever there is a tissue injury which happens and even initiation of cancer is considered as a tissue injury, you have an excess of fibrosis. And when you have excess of fibrosis, it is mediated by TGF beta, and hypoxia and TGF beta signaling synergize in the regulation of fibrosis. TGF beta signaling promotes activation of fibroblasts and remodeling of the ECM. So generally, the local resident fibroblasts will differentiate into the myofibroblasts or the cancer-associated fibroblasts that express the alpha smooth muscle actin. So the alpha smooth muscle actin is a marker for cancer-associated fibroblasts or myofibroblasts. Type 1 collagen, fibronectin and other ECM components. So the fibroblasts will be differentiating into all this. That is myofibroblasts, type 1 collagen, fibronectin and other ECM components. And the TGF beta induced expression of another protein called as the LOX L4P, which is basically a lysyl oxidase like protein. It contributes significantly to type 1 collagen cross-linking, which is increasing the collagen production in that particular area. So the TGF beta and the hypoxia induced the LOXL2P expression promotes collagen cross-linking, leading to ECM remodeling and leading to fibrosis and metastasis. So that is what they have shown in renal cell carcinoma, that is in kidney cancer progression. So you are having TGF beta binding to the TGF beta receptor 1, and this in turn is activating the HIF1 alpha, and this HIF1 alpha can be activated by hypoxia as well. So both TGF beta and hypoxia can induce the HIF1 alpha, and there is a synergy between TGF beta and hypoxia which can lead to renal cell progression, renal cell cancer progression. So the synergy between TGF beta and hypoxia will increase a lot of gene expression like VEGF, leading to growth factor, vascular endothelial growth factor, which means there is more number of blood vessels, GLUT1, then this is carbonic anhydrase 9. All this is increased due to TGF beta, and this will in turn play a role in tumor progression and metastasis. So if you see, TGF beta is such an important player in several cancers, and these are all the genes and proteins which are increased in response to TGF beta. Like in case of kidney cancer, you have an overexpression of CA9, GLUT1 and VEGF. And then all these are important, like CA9 is carbonic anhydrase 9 for pH control, GLUT1 is important for glucose transport, and then VEGF is important for angiogenesis. Similarly, in case of pancreatic cancer, you have fibulin 5, PKC alpha, nestin, NOX4. So these are important for EMT and protection from apoptosis. So kidney cancer, prostate cancer, pancreatic cancer, lung cancer, liver cancer, gastric cancer, colorectal cancer, breast cancer, all these cancers have a collective involvement of TGF beta, promoting invasion and metastasis. These are all the selected genes and in different forms of cancer which is regulated. There is a synergistic cooperation between TGF beta and hypoxia which promotes invasion and metastasis of these cancers. So this is an example to show that this is a pancreatic adenocarcinoma cells. Now this is a tumor environment. So you can see that how this PDAC cells are kind of contained within the tumor microenvironment, and you have all the cells that I mentioned, important cells like you have the TGF beta, and then you have the macrophages, the monocytes, the cancer associated fibroblasts, then you have the antigen presenting happening, the B cells and the T cells recruited. Now you have the myofibroelastic caps, the inflammatory caps, all these are present. What I'm trying to show here is if we are able to block any of these interactions which are happening through certain drugs and agents, then it is possible to stabilize this particular tumor microenvironment, and that is what we will be covering in detail in the next class. So, summarizing the key points, tumors comprise cancer cells as well as stromal compartment with cellular and non-cellular components. The tumor stroma has critical role in cancer development, progression and metastasis. Typically, anti-cancer therapies predominantly target the cancer cells, and their effect on tumor stroma is not taken into account. But it is important to have means to target the tumor environment to facilitate cure. So therefore, the tumor stroma responds to anti-cancer therapies by inducing therapeutic resistance, which can ultimately lead to fatal disease. So it is not sufficient to just address the cancer cells, but it is important to address the tumor microenvironment. The anti-cancer therapies should target both the cancer cells and the stromal compartment to be effective and result in improved patient outcomes. I will repeat this sentence. The anti-cancer therapies should target both cancer cells and stromal compartment to be effective and result in improved patient outcomes. Tumor progression has been recognized as the product of an evolving crosstalk between different cell types within the tumor and the surrounding supporting tissue or the tumor stroma. That is what we saw. What amount of crosstalk happens between the tumor cells and the adjacent cells in the tumor microenvironment. Genetically abnormal cells define the tumor compartment itself. The surrounding and the interwoven stroma provides the connective tissue framework for the tumor tissue. So, the take home messages are: cancer cells can alter their adjacent stroma to form a permissive and supportive environment for tumor progression, and this is known as the reactive tumor stroma. Cancer cells produce a range of growth factors and proteases that modify their stromal environment. These factors can disrupt the normal tissue homeostasis and act in a paracrine manner to induce angiogenesis and inflammation as well as activation of the surrounding stromal tissues. The cell types such as the fibroblasts, the smooth muscle cells, the adipocytes, leading to secretion of additional growth factors and proteases. Activated fibroblasts in the stroma produce tumor progression by secreting growth factors, and the stimulatory migratory extracellular matrix components, as well as upregulating the expression of serine proteases and matrix metalloproteases that degrade and remodel the ECM. The induction of inflammation in the tumor stroma also results in production of a range of factors that produce tumor progression. In a way, these are promoting tumor progression. Angiogenesis promotes not only the tumor growth but also progression from a pre-malignant stage to a malignant stage or to an invasive tumor phenotype. So this neoangiogenesis is a very important area of therapeutic targeting. The tumor stroma can have a more direct role in tumorigenesis and by acting as a mutagen. By normalizing the tumor stroma, it is possible to slow or to reverse tumor progression. So with these take-home messages, we will conclude today's session, and in the next session, we will see how this tumor microenvironment can be targeted through different agents. Thank you.